Nick Majerison here. Welcome to Top Med Talk. Thanks for downloading the podcast. If you like what you hear, make sure you subscribe. This week we're presenting to you our Xtreme Everest mini-series, which we streamed on the website to you live from Duke University in America on June the 14th and the 15th. And this piece we're going to present to you now is called What Happens to Healthy Humans in Hypoxia. It's presented to you by Denny Levett. Have a listen.
Top Med Talk.
Thank you, Monty, for that kind introduction and for the kind invitation to be here in this lovely country, this great institution. What I'm going to spend the next 20 minutes or so talking about is the factors which underlie differential performance at altitude. I'm sure any of you that have been on a high altitude trek or have done any high altitude climbing will have observed is a slightly irritating fact that some people find it incredibly easy, and some of those of us that are slightly less fortunate struggle. It's really frustrating because if you're a struggler, there's very little you can do about it. And if you go and see your doctor and say, what can I do to be better next time I go? The sorry answer is currently get some new genes, or unfortunately what you'll find is each time you go to altitude you may perform in a similar fashion. The curious thing about this is we didn't really understand why that is. And so one of our aims on the Everest expedition was to look at exercise performance at altitude and try and tease out those factors that make people good adapters. Now, as Mike has already alluded to, the apparently obvious hypothesis, or the intuitive hypothesis, is that the reason you can't exercise is that your oxygen delivery has been reduced. And so it's a failure of oxygen delivery, and then we could all go home, and I don't really have much else to say. But actually, as Mike also alluded to, the issue is that if you look at those individuals whose acclimatization response is the best in terms of hemoglobin rise or hypoxic ventilatory response, they don't turn out to be the best performers. And we really wanted on this expedition to look at the variability in performance. So rather than looking at a small group of well-adapted individuals and describing what we see, we wanted to look at a large group of some of whom were previously unexposed to hypoxia and see what it is about those that perform better than others. Now, this is the ascent profile of the cohorts that went on the 2007 expedition, our first large field expedition to Everest. And what I wanted to point out here, you've got days of expedition and altitude here. And the climbers and the investigators are these two here, and the trekkers are dotted line. You can see that the climbers and the investigators took 2 more days to get to Everest Base Camp. Now, Everest Base Camp is 5,300 meters. And in terms of what degree of hypoxia, there's 50% inspired PO2 compared to sea level. So a significant degree of hypoxia. And that was the main difference. The ascent profile within these teams was tightly controlled. Trekkers were not allowed to go on an extra acclimatization walk. They had to sign up to agreeing to that in order to be a subject on the experiment. So we all ascended at exactly the same rate, so that it's a dose-response hypoxia study, essentially. And we're very clear about that and have those ascent profiles carefully recorded for all of the team. And if individuals did actually fall off the ascent profile because they became unwell, then they were excluded from the main analysis. So one of the first things we wanted to look at was this issue of oxygen delivery and oxygen content. So we estimated oxygen content from the 200 healthy volunteers that we took to Nepal by looking at hemoglobin, oxygen saturation, and we ignored the small contribution, which is even smaller at altitude, of dissolved oxygen. So this is clearly an estimation. We weren't going to get ethics to do blood gases on everybody in the field, just those members of the investigator team that felt up to the task. So this is looking at the trekkers, so the 220 healthy volunteers, and the investigators, which were made up by the climbing team and also the laboratory team at Everest Base Camp. And the first thing you can see on the left here, so just to clarify, the white bars is oxygen content on both of these graphs. The dark blue bars here are oxygen sats, and in the light grey is hemoglobin concentration. And if you look at the trekkers, by the time they got to Everest Base Camp, which was 13 days after leaving Kathmandu, they had significantly dropped their arterial oxygen content. In comparison, if you look at the investigator team on the right, that extra 2 days, and we're looking at 5,300A here, that's the arrival at Everest Base Camp, meant that actually by the time we got to Everest Base Camp, our oxygen content was already supernormal. Now that probably reflects the extra 2 days of ascent, and it may be that we're a different group because we're a highly selected individuals that keep going back to altitude, so we clearly perform quite well compared to the unselected trekker group. So we can see that oxygen content and thus oxygen delivery has been reduced in the trekkers and it's been maintained in the investigator team. So what about our performance at Everest Base Camp? So the first thing to say is I led the exercise research on Everest, and these were our aims when we set out. First of all, we wanted to look at changes in peak exercise performance, which we measured with VO2 max, but also sustainable aerobic performance. And for that, we were looking at performance at the anaerobic threshold or the lactate threshold, which I'll come back to. And then we wanted to explore the limitations to peak performance in hypoxia. So what is it that makes you stop exercising when you stop? And finally, we wanted to look at the efficiency of oxygen utilization. Our hypothesis that Mike alluded to was that those that perform well are using oxygen more efficiently, and that underlies their beneficial phenotype that allows them to perform well. And just to prove that I did some of these tests myself, that's me about to start a VO2 max test at, I think that was at Camp 2, and you can see my resting oxygen sats are 70% there. There was an awful lot of work that went on before we left for Everest to make sure that what we measured was accurate and valid. And so the first year and a half of my PhD thesis practically was spent in various types of chamber trying to validate a cardiopulmonary exercise testing piece of equipment to work under those environmental conditions. And that meant a lot of time in chambers with metabolic calibrators. This was actually a meat freezer van that we got donated for 6 months, and we spent a lot of time, as you can see, dressed up in down suits at -20 inside the freezer van. So that I'm pretty confident, and we as a group can be confident, and this data is published, that the equipment was measuring accurately at those altitudes. And not only that, as you can see, the transportation methods in the field were somewhat unusual. And so this is the exercise bike arriving at Everest Base Camp, and we felt that it was probably necessary to revalidate the equipment once we got into our field laboratories so that, again, we could be sure what we were measuring was accurate. And so we had a team with a metabolic calibrator that went up and down all the field laboratories in the 4 locations weekly. They spent the whole 3 months trekking up and down, checking the validity of the kit, so we're pretty happy about that. These are the population demographics, the trekker group on the left, the investigators on the right. You can see about 60% male in the trekkers and 75% in the investigators. And about 60% of the trekkers had never been to altitude previously, whereas the vast majority of the investigators had. So what did we do? Well, we set up, this is the laboratory at Everest Base Camp, and if you go to the new Duke exercise testing laboratory where I spent yesterday afternoon doing our first few patients with John Whittle, it looks rather similar to this. So we had pretty much all the equipment that we would expect to have in a laboratory at sea level. So 2 sets of kit running simultaneously. We did 16 tests a day for 3 months. So we did nearly 2,000 exercise tests. And when my colleagues in the NHS say, oh, it's too difficult to do preoperative exercise testing, I show this picture and say, really, it's not that hard. We did it at 8,000 metres on Everest. It can be done at sea level. So that was more than 90% of what we planned and over 1,000 hours of testing. And what did we find? So then, just to remind you, a reduction in 13% in oxygen content amongst the trekkers. But what about their performance? So this is a VO2 max at sea level, here expressed in mils per kilo, and here in mils absolutes. And this is the reduction as altitude increases. And as you can see, there was a reduction of about a third, so 35% in VO2 peak at Everest Base Camp. So slightly more than the reduction in oxygen content. And what about the investigators? They actually had an 18% rise in oxygen content from baseline. So you would surely expect that their performance was better than the trekkers at least, and perhaps returned to normal because should have normal oxygen delivery. Well, the sorry fact is that actually you see very much the same thing. So look here, this is the investigators on arrival at Everest Base Camp, a 35% reduction in VO2 max, despite normalized oxygen content. And in fact, 3 months later, just before we left, having climbed on the mountain, you can see there's absolutely no improvement in our exercise performance. And just for your interest, at the South Col, which is the highest exercise test that have ever been performed, there was a 61% reduction in peak exercise performance. So despite the fact that oxygen content is normalized, peak exercise performance was extremely limited by about a third at Everest Base Camp. So what about sustainable aerobic performance? So here we are at Everest Base Camp, and here's me with an arterial line in, and these are a series of arterial blood gases. And what we were doing here was validating the use of expired gas to measure the anaerobic threshold at altitude. So the anaerobic threshold is the point in exercise when you do an incremental exercise test where you transition from primarily aerobic metabolism to a greater proportion of anaerobic metabolism. So it gives you an idea of aerobic exercise performance. When you're below your anaerobic threshold, there is very little glycolysis going on. And what happens as you exercise is initially your lactate stays static, and then there's a point here where you can see it starts to rise and never comes back to baseline. And you can see at peak exercise very significant levels of arterial lactate, 14 millimoles. And the lactate threshold, which you estimate with expired gas, is the point at which it starts to rise and doesn't come back to baseline. And as I said, this is sustainable performance. And we validated this measure at Everest Base Camp. I won't go through the detail of those who are not familiar with expired gas analysis, but essentially what this shows is you can identify the lactate or anaerobic threshold accurately, even with the presence of additional hypoxic drive, which you have at Everest Base Camp. So what happened to the anaerobic threshold? Well, here we have the trekkers. There's a reduction of 30% in your aerobic performance at Everest Base Camp. And when we look at the trek and the investigator team, you can see that it's about 28% that your anaerobic threshold reduces, and that at the end of the expedition fell slightly further. So actually what we found, which is a novel finding, is that altitude seems to affect your peak performance, perhaps counterintuitively, more than your aerobic performance. So somehow we are actually maintaining our aerobic performance better than our peak performance, although both are reduced at Everest Base Camp. So to summarize in performance terms, acclimatization returns oxygen content to normal, but performance is still significantly impaired. So then we went on to look at what was predicting the decrement in performance amongst both the trekkers and the investigators. And the first thing we looked at, because it seemed sensible, was the change in peak performance, so the reduction in VO2 max, and we express that as a percentage, because clearly if you don't express it as a relative fall, then the fit people will have a greater fall, versus oxygen content. And this is either resting oxygen content or oxygen content measured at VO2 max, because when you exercise at altitude, you do desaturate. And if you look at either of those measures, what you can see, I don't think you have to be a statistician to see that there is basically no relationship between those two variables. So oxygen content variables do not predict exercise performance at extreme altitude. Age also, as you can see, doesn't predict exercise performance. So for those of you who in your retirement want to go to Everest Base Camp, there's no reason why you shouldn't, because you won't perform any worse than you did when you were younger. And equally, gender doesn't affect your performance. In fact, when we look through a huge number of variables, the only thing that had a very significant effect on performance was your baseline fitness. And for those of you that perform poorly at altitude, you can reassure yourselves that actually the fitter you are, the worse you do at altitude in terms of decrement in exercise performance. So that's clearly why I'm always trailing my husband when we go there. So what is it? If it's not oxygen content, what is it that's limiting exercise performance? If you ask a subject at Everest Base Camp why they stopped exercising at the top of an incremental test, they will all tell you it's dyspnea. They can't breathe. If you ask them in the lab at sea level, they'll tell you it's their legs. In fact, if someone at sea level tells you they've got dyspnea as a main reason for stopping exercise, that's a key sign that you need to go and look for a problem either with cardiorespiratory function, because that's an abnormal reason for stopping exercise. But that's what all of the subjects will tell you at VO2 peak. And so there had been a suggestion that one of the problems with exercise capacity is because your respiratory rate and your minute ventilation are so high, actually you're hitting your maximal voluntary ventilation, and the reason that you stop exercise is you have no breathing reserve. So we measured this directly. There have been a couple of studies previously looking at this with estimated maximal voluntary ventilation using FEV1 as an estimator. The problem is that changes at altitude because of the reduced density of air. So that was suggesting that breathing reserve fell, but when we measured it directly, what we found in red here, so in green you've got maximum voluntary ventilation, you can see with altitude it actually rose. And that's probably because of the reduced density of air, so it was less work of breathing. And so as a result, actually breathing reserve increases. And so breathing reserve at Everest Base Camp was higher than breathing reserve at sea level, and so certainly couldn't be thought for the reason for why individuals stopped exercising. And it comes back to a really important point, that the sensation of dyspnea does not necessarily relate to the physiological measures of dyspnea. So your PaO2, and we know that from clinical practice, the COPD patient that's a blue bloater as opposed to the pink puffer, that's because some people are very tolerant of hypoxia. So some individuals who are really very hypoxic don't actually get very much dyspnea, whereas others do. One of the other suggestions has been that the reason that you stop exercising is that you, at the last point, defend your cerebral saturation. And so the hypothesis is that those who stop earlier are individuals whose cerebral blood flow falls earlier, their cerebral sats drop first, and then you stop exercise because you're defending your cerebral sats. We use near-infrared spectroscopy, which is an indirect index of cerebral oxygenation, and found quite clearly that there was actually no relationship between your cerebral saturation either at rest or anaerobic threshold, or indeed just before peak exercise. It certainly didn't predict those who decremented their VO2 max more. So looking at the South Col test, so this is one of these 5 tests we did on the South Col, powered by car batteries, which the Sherpas lugged up the mountain for us. We just about managed to pull ourselves up the mountain. And what you can see, those of you who probably are not that familiar with looking at expired gas analysis, I'll just point out a couple of things on this graph. First of all, here's ventilation in litres a minute. You can see at peak exercise, 200 litres a minute at the South Col. The second thing is over here, look at your end-tidal. These are end-tidal gases. So resting end-tidal CO2 down at about 12 to 13 millimetres of mercury. Those of you that are familiar with exercise testing and know how to identify the anaerobic threshold, here is the V-slope. And although the numbers are very abnormal, what's really remarkable about this graph is how the patterns are exactly as you would expect them to be at sea level. So we were really pleased with the quality of the data that we got. And you can see a very clear inflection in the V-slope here, which marks the transition to anaerobic exercise and more expired CO2 measured at the mouth. But what's really interesting is what's going on in this graph over here, which is heart rate against work rate during the test, and something called the oxygen pulse, which is a surrogate measure of stroke volume. And what you can see, you would expect this to be increasing throughout the test. And what you see is here, there's a clear inflection point at this point here where your heart rate shoots up as a compensatory measure to maintain your cardiac output. And the reason you need to do that is because you can see this oxygen pulse is completely flattened, which indicates that at that point there was a real problem with stroke volume incrementation or AV extraction. And so what it looks like here is at the South Col, our investigators were actually limited by cardiac function. So if we go back to the oxygen cascade, and if you remind yourselves of this, what we have after the systemic circulation is a microcirculation, the mitochondria. And what I've been talking about up to this point is about arterial oxygen content, about cardiac function and systemic oxygen delivery. But actually what we've seen is that there seems to be a dissociation between central oxygen delivery and what the tissues are actually seeing. And so clearly it appears that the deficit in performance is caused by something that's happening distal to the systemic circulation. So oxygen content is normal, cardiac output is maintained until extreme altitude, but what we're finding is that performance is still impaired. And so we hypothesized that the problem may be a metabolic problem, and what was happening may be a change in the utilization of oxygen in the tissue. So our other exercise experiments were looking at the efficiency of oxygen use. And if you look at exercise in the aerobic portion, so below the anaerobic threshold, so easy, sustainable exercise, not hard work, and you get individuals to repeat the same exercise challenge at sea level and in different degrees of hypoxia, you can look at how much oxygen you're using, the oxygen cost of a given amount of work. And what we found is, so here we've got the work rate in watts, so 20, 40, 60 watts, very low work rates, and this is the oxygen uptake to perform those work rates. And I think you would agree with me that in some ways I was pleased about the data quality, because this is London, Namche Bazaar and Everest Base Camp, but those lines very much overlie each other. So this is in the trekkers, and what you can see is that there is absolutely no change in the oxygen utilization or the oxygen cost of work over the course of the altitude ascent. Interestingly, however, if you looked at the investigators, their early data was very similar, and there was no change in oxygen utilisation efficiency. But by the end of the expedition, you can see that there is a clear reduction in the oxygen cost of the same degree of work. So suggesting that there was actually a change after prolonged altitude exposure. And this led us in 2013 to want to go back and to look at the indigenous Sherpa population, who are perhaps, in terms of evolutionary exposure, the high-altitude native population that have been exposed to hypoxia for the longest evolutionary time, and to look at the efficiency of oxygen utilisation in the Sherpas to see if this underlay their extremely good performance in hypoxia. And so what did we find in the Sherpas? Well, I tell you, the first thing we found is that Sherpas find it very difficult to cycle because they didn't actually cycle when they were children. So I spent most of the first few weeks of this experiment teaching the Sherpas how to cycle. It was very difficult because if you talked to them or distracted them, their feet would fall off the pedals, and we ended up having to tape their feet to the pedals to get them to be able to cycle. It's interesting, even with elderly patients, you don't have that problem. And I think that's because most of us in the Western world have cycled at some point or used a pedal bike during our childhood, so that motor memory is firmly still present. But having got over that challenge, we looked at their oxygen utilisation uptake. And so this is in Kathmandu, because the baseline was Kathmandu for the Sherpas. Again, Namche Bazaar in orange and Everest Base Camp in grey. And what you can see is that the Sherpas, unlike the lowlanders, had a clear reduction in the efficiency or the oxygen cost of a given level of work. Again, which would indicate that perhaps what underlies their performance characteristics is that they are using the oxygen that they have available more efficiently than lowlanders. So in summary then, what I've described is the fact that systemic oxygen delivery is preserved until extreme altitude. Both maximal and moderate intensity exercise, even at relatively moderate altitudes, despite normalization of the oxygen delivery, remain impaired, which suggests that there is either impaired oxygen delivery distal to the systemic circulation, or there are changes in the utilization of oxygen at a cellular level. And there is some suggestion from my work, and I'm going to hand over now to other colleagues who did some work looking more at the metabolic changes and the microcirculation that may underlie the good performers at altitude, the star altitude performers, and perhaps even the star performance that we see in the Sherpa population. I don't think I should end, however, without mentioning our Sherpa team. We were really fortunate to have the same team when we went on a previous 2 expeditions, in fact, to Choi Oyu, which is another 8,000 metre mountain in Tibet. So by the time we got to Everest, it was the 3rd expedition with the same Sherpa team. And these are 4 brothers and their nephews. And they are an amazing band of brothers from the Rowling Valley. And this is Pema here on the left, was our base camp Sirdar. So he was in charge of arranging everything for the expedition, research and supplies at base camp. But he was a slight embarrassment to his other brothers because he was the only one of the 7 that hadn't summited Everest previously. So in fact, on our expedition, we treated him as one of the clients, and on the summit day, he went up with an extra Sherpa and an extra bottle of oxygen, and he summited along with Mike and Dan and Chris in the front row. And I'm very pleased to announce that just this year, in fact, Pasang, who's one of the brothers here, and who also carried the blood gases down from the summit, they've all just received this Guinness Book World Records certificate for the most siblings on an 8,000 metre peak, which was 4 of them simultaneously in 2007, along with Mike, Dan and Chris, which is really fantastic. But that actually is only a small idea, a small part of their amazing achievements as a group of elite altitude performers, because they got 2 other world records at the same time. So the most siblings to have climbed Everest is 7 by Nepalese brothers, so all of them have climbed Everest. Sadly, only 6 of them survive, and the one that isn't alive didn't die climbing but had medical problems. But look over here. Between them, they've done 61 ascents of Everest. I mean, just quite extraordinary. And Mingma, who is this chap here, who was our climbing lead, has also, amongst other 8,000 metres, he's climbed over 30 himself now on top of Everest, including K2, which in climbing terms is an entirely different challenge. So thank you very much for listening, and I'd like to thank all of our collaborators and funders.
Don't forget you can meet the Top Med Talk team, ebpom.org/meetings. Our next big event is on the 28th to the 30th of September. EBPOM USA Chicago Masters Course, a perioperative care practicum. Full details available on the website ebpom.org/meetings. I'll see you there.